This is the metabolic poster pathway presentation. It's to show how the body can generate ATP and otherwise the building blocks of DNA. Starts with the glycolytic pathway. Our starting materials are glucose and carbon dioxide. Glucose enters the glycolysis pathway and then is broken down into pyruvate. This consumes ATP, produces 4-ATP along with 2 NADH molecules to continue on. Pyruvate is then converted to acetyl-CoA via the pyruvate dehydrogenase complex. And this is in order to commence the citric acid cycle. Pyruvate is converted to acetyl-CoA via pyruvate dehydrogenase complex, where another NADH is produced. Pyruvate can then enter the citric acid cycle, where 3 more molecules of NADH are created along with one of FADH and FADH2, along with a GTP molecule. These electron carriers, NADH, FADH, FADH2, then donate their electrons along the electron transport chain. This causes them to oxidize into NADH plus and FADH plus. And as the electrons move downward along the electron transport chain, the protons are shunted from the inner membrane space. Shunted from the outer to the inner membrane space. These protons return to the mitochondrial complex via ATP synthase, where they are accepted by oxygen atoms producing water. ATP is also produced in this process by combining ADP and inorganic phosphate. Back in the glycolysis process, a molecule called glucose 6-phosphate, or G6P, is produced via the enzyme hexokinase. And G6P then enters the pentose phosphate pathway, where it is converted to ribulose 5-phosphate, which is then converted to ribose 5-phosphate and xylulose 5-phosphate. R5P is then converted to PRPP, where PRPP helps to synthesize uridyl and inositol. And then uridyl is then converted to either UTP, CTP, TTP, which are pyrimidines, and inositol is either converted to ATP or GTP, which are purines. These resultant nucleotides made from this process and then used for DNA or RNA synthase. And this last slide is to sort of illustrate how certain nucleic acids can then be reconverted or rebroken down back into certain materials, which can then enter the metabolic pathway at certain points, again, like pyruvate or oxaloacetate or ketoglutarate.